Good morning, students. Hope you be fine at your respective places. Today, we are going to discuss the structure of the DNA. Structure of this DNA. Now, before going in detail about the structure of the this DNA, let us go towards the historical perspective of this DNA. Now, this DNA, what we will see, it is a nucleic acid. DNA is a nucleic acid and it was first of all reported in past cells by Frederick Mesher. This DNA was first of all reported in past cells by Frederick Mesher. Also this DNA, it was reported as acid. What we call it, this was reported as acidic substance, rather as an acid or acidic substance in nucleus by the same very person. Second, जो हम बात इसकी कर लेंगे रिगार्डिंग दिस डीएनए was first of all जिसकी टर्म है द टर्म अगर हम बात कर लेंगे दिस was first of all given by that is called as a Zacharias. And the model अगर हम कहेंगे the structure rather of this DNA, it was given by Watson and Crick. So regarding the historical perspective of this DNA is a nucleic acid. It is a DNA molecule, is a DNA, this nucleic acid, and it was first of all reported in past cells by Frederick Mesher, and he also reported it as an acidic substance in nucleus. The term of this DNA, it was coined by first of all by Zacharias, and the structure of this DNA, it was given by the Watson and the Crick. एक और बात जो हम कहेंगे DNA अगर हम बात कर लेंगे it is a polymer. That means it consists of many molecules. It is a polymer of nucleotide, and each nucleotide की अगर हम बात कर लेंगे it is made up of pentose sugar, and the pentose sugar here it is deoxyribose. It consists of nitrogen base, and these nitrogen bases, they may be of two types. First one is the purine, and second one is the pyrimidine. In case of DNA, out of two purines, both of them are present, and out of three pyrimidines, only two are present in this. That is, except uracil, and that uracil is found in the RNA, and then there is the phosphate group. So, what we will see this DNA is a polymer of nucleotide, and अगर हम देखेंगे जो न्यूक्लियोटाइड का जो हम देखेंगे इट इज स्ट्रक्चर इट इज द पेंटोज शुगर विच इज मेनली द deoxyribose, and there is a nitrogen base. That nitrogen base may be two purines and two pyrimidines, and there is the phosphate group. If we draw the structure of this nucleotide, if we draw the structure of this nucleotide, you will see This is carbon first, this is carbon second, this is carbon third, this is carbon fourth, and this is the carbon 5. To this carbon 5 is attached the phosphate group. On this carbon first we have the nitrogen base. And again what we will see there, in case of this DNA, here we will see it is the hydrogen bond and hydrogen bond. Here we will see at carbon third it is hydrogen and hydroxyl group, and here what we will see it is the hydrogen bond. So what we will see here, this the difference between the ribose sugar and deoxyribose sugar is here at C2 group. In case of the ribose, it is the hydroxyl group, while as in case of the deoxyribose nucleic acid deoxypentose sugar, it is the hydrogen group. Now, the what we will see जो nitrogen base है, हाँ जो अब कहेंगे जो दूसरा, when it forms the chain of these nucleotide, the next nucleotide जो हमारे पास है will form a what we call as the bond, and that bond is called as phosphodiester. That is called as a 5 phosphodiester bond that is found, and the next जो nucleotide that will get attached with this nucleotide at this hydroxyl group, which is found at the third carbon. And the bond between this carbon first and the nitrogen, it is also called as glycosidic bond. This is called as the glycosidic bond. Now nitrogen base जो है कि how it is what we call as attached with the carbon, depending upon what we will see the nature of nitrogen base in this in what we will see, if we will see depending upon the nature of the nitrogen base. In case of purine, अगर हम प्यूरीन की बात कहेंगे, दिस कार्बन फर्स्ट जो है इट इज अटैच्ड एट 9th, what we call as the 9th position, of this purine. And in case of pyrimidine, it is attached at first position of the pyrimidine. So depending upon the nature of the nitrogen base, how it is bonded with this carbon first, in case of purine, this carbon first is attached with the 9th position of the purine, and in case of the pyrimidine, this carbon first is attached at the first position of the pyrimidine. Now, दूसरी बात, the second, what we'll see this, what we call as this pentose, what we call as this nucleoside, what we will see, this nucleotide will form a bond with this hydroxyl group, which is attached at the third carbon by phosphodiester bond. This is the structure. Now जो हमारे पास the structure which has been given by the Watson and Crick, let us try to discuss about that. The bond which has been, the structure which has been given by the Watson and the Crick, and that is simply called as the double helix. Double helix structure of DNA. See what Watson and Crick, and it is simply called as the Watson and Crick's model of DNA, or we can simply call it as a Watson and Crick's double helix structure of DNA. What they have said, number first point, that उन्होंने कहा DNA helix जो है, it is a double stranded. It is a double stranded. So we write it as DS. DNA helix, it is a double stranded. Second one, each strand is made up of, हर एक स्ट्रैंड जो है इट इज मेड अप ऑफ न्यूक्लियोटाइड्स. Each strand is made up of nucleotides. Third, the two nucleotides. डीएनए हेलिक्स इज ए डबल स्ट्रैंड. जब हम डायग्राम बनाएंगे वो फिर समझ आ जाएगा. ईच स्ट्रैंड इज मेड अप ऑफ न्यूक्लियोटाइड्स. थर्ड बात जो हम कहेंगे, द टू न्यूक्लियोटाइड्स आर anti-parallel to each other. What does this mean? We will try to discuss it. Or anti-parallel to each other. That is the third point. First point, DNA helix is a double-stranded. Second, the each strand is made up of nucleotides. Third part, the two nucleotides are anti-parallel to each other. Now, let us talk about what the things exactly mean. I have given you the 3 points. What these 3 things exactly mean, or how we can say, or we can, I mean to say, how the structure of this DNA is going to be. Let us compare the structure of DNA with a spiral staircase. Let us compare the structure of this DNA with the what we call as spiral staircase, like this. अब जो इसकी these are staircase, और जो ये railings इसके है, this is the one side railing, is another railings. These are the strands, and these strands जो हम कहेंगे, these railings जो है, they are parallel to each other and they are anti-parallel to, we simply say they are parallel to each other and they do not cross each other. These railings, I compare the structure of DNA with this spiral staircase, and these are the railings. This is one railing and this is second railings, and these railings, they are parallel to each other and they do not, what we will see, intersect with, or they do not meet with each other. Now if I try to draw the diagram, see here, जो स्ट्रक्चर इसका बन जाता है डीएनए मॉलिक्यूल का, these are parallel to each other. They do not cross with each other. And they do not cross with each other. this is somewhat the structure of this. This is the double helix, one, and this is the second. This is a double strand. This is the one strand, it is the second strand. Now each strand is made up of nucleotides. There are the nucleotides. How these are arranged, I will show them. Now the two nucleotides are anti-parallel to the two nucleotides. They are anti-parallel to each other. What does this term anti-parallel means? For example, if in one strand the third carbon, for example, in this strand the third carbon, if in one strand the third carbon is free, and in the other end it would have, what we will see, in the other end it would have the 5 carbon free in the same strand. इसका एक हैंड है, दूसरे हैंड में हम देखेंगे देयर विल बी द 5 कार्बन फ्री. रखा याद. एंड वी कॉल्ड इट एज 3 prime and 5 prime, or we call it a 3 and 5 end. For example, if we see, if one strand, this is the strand, if in one strand, what we will see, the third carbon is free, that is at one end the third carbon is free, and the other end of the same strand, अभी दूसरा strand इसी का है, where we will see the 5 carbon is free, so we called it as a 3 prime and 5 prime, or we call it as a 3 and 5 end. Now parallel simply means, parallel means that these do not cross with, this is a one strand, this is the second strand, but these two strands, they do not cross with each other, and they are equidistant from each other. They are equidistant, and the distance between them, the two, that is same. Whatever there is a top distance between these two strands, and whatever the distance is at the bottom, that is the same. Now, what do we mean by the anti-parallel? Anti-parallel means if in this strand the free carbon atom, जो हम देखते हैं यहाँ पर जो free है, that is the third carbon. Now, the strand which is in front of it, that is this strand. अब जो यहाँ पर आ जाएगा, I will write it. It will be the 5 carbon free, and जो इसका bottom होगा, that will be the 3 carbon. क्योंकि यहां पर जो हम देखते हैं पहले स्ट्रैंड में, जो पहले स्ट्रैंड में है, here it is the 5 carbon, and here now it will be the third carbon. Now let us see a detailed structure. I will try to draw the diagram. Let us try to let us try to see a detailed structure of this anti-parallel nature. Say we have a, what we call as a nucleotide. Let us pentose sugar हम बनाएंगे. See here, pentose sugar, this is carbon first, carbon second, this is carbon third, this is carbon fourth, and it's the fifth. This all. Now what we will see with this carbon 5, we have this phosphate group. I write this. We have got this phosphate group. Now at this carbon first we have then nitrogen base, and this is जो bond है carbon और nitrogen, that is the glycosidic bond, and this phosphate, it is bonded with this fifth carbon by means of a phosphodiester bond. Now what we will see in case of this C3, what we will see, there is OH bond, and under some देखेंगे H and OH, and this is the H and H. Let us try to draw another one, pentose sugar, this nucleotide. This is carbon 1, this is carbon 2, this is carbon 3, this is carbon 4, and this is carbon 5, C5, and this is phosphate group. Now this phosphate group is attached with the C3, third carbon. This phosphate group of this nucleotide, it is attached with this phosphate. So here what we will see, in this case the OH, what we will see, this functional group is free, OH, that is there, and at stop it is the fifth carbon which is free. Now in the second strand what we will see, upward down हम कर लेंगे structure. I will draw this with this. Take a minute. C1, carbon first, carbon second, this is carbon third, this is carbon fourth. See here. Now here we have got the nitrogen base. This is the nitrogen base, this is the nitrogen base. Here we will see it is H and H. Here we will see it is H and it is hydroxyl group. Now this is this what we call as the carbon fourth, and this is the carbon fifth, C5, and it is the phosphate, P. Now we will try to draw another one. C1, carbon first, this is carbon second, this is carbon third, this is carbon, and here it is hydrogen and it is hydroxyl. This hydroxyl bond will be attached with this phosphate group. There it is the hydrogen and hydrogen. Now here we will see it is hydrogen again. Now here it is the nitrogen base, this nitrogen base, and again there it is the nitrogen base. Here nitrogen base, थोड़ा बहुत ऊपर की ओर खींच लेंगे. It is the nitrogen, sorry, this is carbon first and it is the nitrogen base. Now here we will see what is there. जो free है that is the C3, it is this strand, and जो free हमारे पास है this is this carbon fifth, and this is this carbon fifth जिसके पास phosphate group है. So what we will see in this first strand, we will see we have got fifth carbon free here, and one and other end it is the third carbon that is there, fifth carbon here and third carbon here. In the second we have got this third carbon free and fifth carbon free. See here, third carbon and the fifth carbon free. Now one more कि these nucleotides, अगर हम बात कर लेंगे इन न्यूक्लियोटाइड्स की, हम बात कर लेंगे एक और बात यहाँ पर हम ये देखते हैं कि that these two nucleotides, the two nucleotides are attached. The first nucleotide जो हम बात कर लेंगे it as hydroxyl group free and phosphate at that is at carbon 3, and second at phosphate at carbon 5. दूसरे न्यूक्लियोटाइड अगर हम यहाँ पर देखते हैं कि इस स्ट्रैंड में पहले हम क्या देखते हैं कि it has got third carbon free दैट इज हैविंग द फंक्शनल ग्रुप हाइड्रोक्सिल ग्रुप। और सेकंड में हम देखते हैं कि फिफ्थ कार्बन जिसके पास फॉस्फेट ग्रुप है जो हमको यहाँ पर दैट दिस मीन दिस इज द एंटी पैरेलल। अब यहाँ पर फिफ्थ कार्बन फ्री है और इसके सामने वाला जो है न्यूक्लियोटाइड वहां पर थर्ड कार्बन फ्री है। अब यहाँ पर थर्ड कार्बन फ्री है, इसके सामने जो हम देख इन फ्रंट ऑफ दिस ट्रेन दिस इज द फिफ्थ कार्बन फ्री। दूसरी बात, नाउ देयर आर द बेसिस, दीज आर द नाइट्रोजन बेसिस। नाइट्रोजन बेसिस की अगर हम बात कर लेंगे हियर व्हाट वी मीन वी टॉक ऑफ द नाइट्रोजन बेसिस, दीज नाइट्रोजन बेसिस what we will see we have got two types of nitrogen base. First one is purines and second one is pyrimidines. These purines and pyrimidines की अगर हम बात करेंगे they pair with each other. This purine it pair with the pyrimidine. I will write pyrimidine for py. Now the pairing will be adenine it pairs with this thymine by means of the double bond. These are the hydrogen bonds but these are the weak bonds. दूसरी बात जो हम कहेंगे guanine it this cytosine by means of the triple bond. Both these bonds जो हम बात करेंगे they are hydrogen bonds but these bonds are somewhat weak. Now what will happen, for example, if here the nitrogen base is adenine it pair with this base that is the thymine, this thymine with the help of what we called as the double bond. Now if here the nitrogen base is what we'll say guanine it will with cytosine with the help of triple bond. So this is how these bases are found. Third बात, अगर हम बात कर लेंगे what we will see if we will draw this axis, if we try to draw the axis like rather this we will see जो diameter इसका होगा diameter it has got the uniform this double helix structure DNA it has got the what we call as the uniform diameter and that diameter की अगर हम बात करेंगे it is 20 angstroms. जो हम एक और बात अब फोर्थ बात हमारे पास आ गई जो डायमीटर हमारे पास है that is जो इसका डायमीटर है that of DNA is 20 angstroms. जो डायमीटर है इट इज 20 एंगस्ट्रॉम. फोर्थ बात, हाँ बस अब जो हम ये कहेंगे दैट दिस इज पॉसिबल बिकॉज अब उसकी पॉसिबिलिटी क्या है व्हाट इज दैट पॉसिबिलिटी दैट द डिस्टेंस बिटवीन दीज एडेनिन एंड थायमीन एंड हम कहेंगे बिटवीन टू नाइट्रोजन बेसिस दैट इज सेम. सो दिस double helix structure it has got the uniform diameter because the distance between these nitrogen bases यहाँ पर हमने देखा adenine and thymine या guanine and cytosine that is uniform. जितना यहाँ पर distance है उतनी यहाँ पर भी spacing है. So this has got the what we will call as the uniform distance or diameter from top to the bottom. Second, what we will see one turn, फिफ्थ बार वन टर्न जो हम बात करेंगे वन टर्न ऑफ डीएनए इज इक्वल टू 34 एंगस्ट्रॉम्स. नाउ एक टर्न हमारे पास कितनी है आई विल शो दिस. इफ वी हैव गॉट द स्टेयरकेस it is the roundest staircase, we have got the spiral staircase rather. जो हम इसका पहला स्टेप होगा when we ऊपर चले जाएंगे एंड वी विल reach at that staircase at that what we call the step which is parallel to this which is above this first rather हम कहेंगे वो consecutive step जो है which is above this first step that will form the one turn. In case of this what we will see in this what we will see this is somewhat narrow groove. बट जब हम यहाँ की ओर चले जाते हैं, दिस इज समवट डीप ग्रूव. यहाँ की ओर इट इज समवट नैरो ग्रूव. अब फ्रॉम वन नैरो ग्रूव टू दिस, जो डिस्टेंस इसकी आती है यहां से यहां तक, दिस इज द वन टर्न. That one turn is equal to 34 angstroms. Now, in this groove, what we will see, there are about 10 nitrogen bases, 10 pairs of, in this one turn, there are about 10 pairs of nitrogen bases. And so what is the distance between the two consecutive nitrogen bases? That is simply 34 divided by 10 is equal to 3.4 angstroms. So one turn is equal to, one turn of this DNA helix is 34 angstroms, and in each turn there are about 10 pairs of nitrogen bases. And what we will see, जो अब distance between the two consecutive nitrogen bases, that comes to equal to 3.4 angstroms. एक और बात, अब जो यहां पर हम बात कर लेंगे, Watson and Crick, they have also given us, Watson and Crick, एक और बात उन्होंने बताई, that there are 5 types of DNA. Watson and Crick, they have given us that there are the 5 types of DNA. These are DNA A, DNA B, DNA C, DNA D, and DNA Z. Watson and Crick, they have also given us different types of the DNA, and they have given us the 5 types of the DNA. They are DNA